Welcome back to our Science ACT overview. Today we are going to look at a research summaries passage and talk about the kinds of things that you'll see and the kinds of questions that they'll ask when you come to these passages. These passages often look at multiple experiments that are trying to study one thing, and the questions that you might see may ask you to analyze the design of the experiment, predict possible outcomes if you change certain variables, determine a conclusion for the whole study, or simply just to identify information. As you are going through these passages, make sure that you're paying attention to a few things. Notice any changes in the experiment that you may see when they talk about each particular study. Also notice what is the control variable in each study. And pay attention to changes in percentages, concentrations, speeds, or time frames that are listed along the side. Before we look at the passage, let's review the types of questions you may see. This is very important for you to remember because you want to know what a question is asking you when you actually see the question, and the best way to do that is to identify the type of question that it is. Again, we have detail questions, asking about specific details. Pattern questions, asking you to identify trends. Inference questions, asking you to draw conclusions on the information given. Scientific method questions, asking you to examine the experiment itself. And compare and contrast questions, asking you to compare viewpoints or experimental data. Okay, so I'm going to read through the passage once, and then we're going to go back through the passage and look at the information that is provided in each section. Here we go. Yeast is a unicellular fungus and is arguably one of the most important members of the fungus family, primarily because of its involvement in the process of fermentation. During this process, yeast breaks down sucrose into alcohol and carbon dioxide. The chemical equation for fermentation is given below. Scientists study how to induce fermentation in yeast most effectively. Experiment 1. Since yeast needs sucrose to ferment and molasses is 60% sucrose, scientists first study yeast grown in molasses. 5 test tubes are filled with 0.4 grams of yeast and various molasses concentrations. Carbon dioxide levels are measured as an indication of fermentation for each test tube after 1 day and again after 2 days. These levels are shown in Table 1. Experiment 2. 5 different test tubes are filled with 0.4 grams of yeast and various pure sucrose dilutions. Carbon dioxide levels are then measured as an indication of fermentation after 1 day and 2 days. These levels are shown in Table 2. Experiment 3. 8 more test tubes were filled with 0.4 grams of yeast and 15% sucrose solution. Various combinations of ammonium sulfate, potassium dihydrogen phosphate, minerals, and vitamins were added to the test tubes. Carbon dioxide levels were measured after 1 and 2 days. These levels are shown in Table 3. Now, that's a lot of information that's been given to you in this passage. You're not expected to fully understand what is happening in this experiment. What you need to do is get the gist of what we're looking at. For instance, we know that yeast is being used in this experiment to look at fermentation. And we're going to be looking at sucrose. In experiment 1, we see that we're dealing with molasses, which is 60% sucrose. Pay attention to that 60% sucrose. That will come into play later. We also know that we're looking at various molasses concentrations, and we're measuring carbon dioxide on day 1 and day 2. Notice off to the left, it's showing us our concentration for molasses for each experiment. Pay attention, this is percent molasses, not percent sucrose. I've also included 2 arrows to show my trend from smallest number to biggest number. This shows me that my percent molasses and my CO2 concentration is directly proportional. For experiment 2, I'm looking at pure sucrose dilutions. Again, I'm going to draw my arrows from smallest to largest to see, one, that they are directly proportional, but notice that I'm starting with a higher percent concentration in this experiment than I did in experiment 1. In experiment 3, you can see that our concentration of sucrose remains the same, but instead I'm going to have various combinations of other materials that I add to my sucrose. All of those combinations are listed here. Now let's look at the questions that you may see in this type of passage. Note that a lot of questions may ask you to look at information from more than one experiment, so pay attention to what you are looking at. Here is question 1. Based on experiment 1, which test tube showed the highest level of fermentation after 1 day? This question is a detail question. It's asking you to go back to experiment 1, to look at day 1, go down and find the highest level. For us, that's 86. That happens in test tube 5. If I come back to the question, very easy, I see test tube 5. Question 2. Which of the following variables was controlled in experiment 1? This is also a detail question. It's asking you just to look at the information from experiment 1 and figure out what did you keep the same. If I look at my experiment, I can see that I am changing the amount of molasses for every test tube. But if I look back up in the passage, I can see that what I'm holding constant is the amount of yeast. If I come back to my question, I'm given the answer choice F, the amount of yeast is held constant. Question 3. Which of the following is a weakness in the design of experiment 3? This is a rare scientific method question. These types of questions are asking you to analyze the strengths and weaknesses of the experiment. Because this is just an overview of the passage type, I'm going to go straight to the answer. Here in experiment 3, I see that I have various combinations of other materials. I will tell you now that the answer choice is D. If I look at the graph, I see that there are lots of materials that I can use, but I'm not seeing all the combinations. For instance, I don't see ammonium sulfate combined with vitamins. Here, if I go back to my question, I see that answer choice D is telling me all possible combinations were not tested. Now, in future videos, I'll go through each of the answer choices and explain why they're not correct. But because this is just an overview video, we will move on. Question 4. Based on experiments 1 and 2, one can infer that as compared to molasses, pure sucrose is blank. This question is an inference question. It's also asking you to compare multiple experiments, something that I pointed out before that you might have to do. Let's look at our answer choices to see what this question is really asking. Answer choice A is more effective at inducing fermentation. Answer choice B is less effective at inducing fermentation. C is exactly as effective, or D does not induce fermentation in yeast. When we look at our experiments, we'll need to pay attention to the levels of fermentation. So here we have experiment 1 and experiment 2. Now, this is a little tricky, and this is why I was telling you to pay attention earlier. Notice here that this says 60% sucrose for molasses. So, if my percent molasses is 100%, that means that my percent sucrose is only 60%. If my molasses is 50%, my sucrose is 30%. My molasses 25%, then my sucrose is 15%. This is a trick that test writers put in here to see if you're really paying attention. I can't make a comparison unless I know how much sucrose is in each experiment. Once I know how much sucrose is in each experiment, I can begin to make comparisons. Notice here I have my 60% sucrose. The numbers don't seem that terribly different, 86 or 90 on day 1 and 100 on both day 2s. Here I see 69 and 94, but I see 78 and 100 for pure sucrose, something that looks a little bit higher. For 15% with my molasses, I see 24 and 78, but with my pure sucrose at 15%, I see 40 and 84, leading us to believe that pure sucrose is actually more effective at fermentation than sucrose coming from molasses. If I go back to my question, I can see that answer choice A is saying that pure sucrose is more effective at inducing fermentation in yeast than is molasses, which is the conclusion that we just drew from our charts. Question 5. In order for scientists to decide on which conditions produce the most efficient growth of yeast, which of the following experiments would most likely need to be completed next? This is another elusive scientific method question. In this question, they're asking you to look at the experiment as a whole and decide how do you proceed from this point. Again, because this is just an overview, I'm going to go right to the answer. In experiment 1, we not only looked at varying our amounts of sucrose, but also our source of sucrose. In experiment 3, we looked at all of the added materials that we could include in our sucrose mixture. The only thing that we have not changed is the amount of yeast that we have used. So if we go back to our question, we will notice that answer choice F tells us that we can use a varying amount of yeast to take our experiment beyond what we've looked at thus far. Now, because this is an overview, I went straight to the answer. However, in future videos, again, I will go through and I will explain why each other answer choice is incorrect. Just to recap, when you see a research summaries passage, make sure that you're paying attention to each of the studies presented and the variables that change between each study. That's all for today. Tomorrow, we'll look at a conflicting viewpoints passage.